really at the end of Bill's talk, which is the cause or consequence question. I think we should add on Norbert's additional question, is that the wrong question? And we wanted to invite physician scientists to try to address this both from their clinical perspectives as well as their basic research interests. So I thought maybe we could start by having each of you tell us a little bit about where you're coming from and kind of how you think about these questions.
So where I come in on this as a transplant immunologist, okay, so when I actually started off and I did my K award in a diabetes islet transplantation project and moved across the street in Colorado to when Ruben and Norbert were working on immunity, and they said, we're seeing all this inflammation, what does it mean? And I said, I don't know. But in the autoimmunity world, in type 1 diabetes, and certainly in transplantation, we don't torture ourselves wondering if the immune response is contributing. So because it usually, it's fairly well accepted. And the other thing that I kind of alluded to with Edda earlier is that we don't, with other autoimmune diseases where the blood vessels are involved, we don't question when we see inflammation that's what's going on. So anyway, to cut a long story short, when we got into the athymic rat model and we realized that the absence of T cells didn't make it better, it made it worse. And when the inflammation was worse, and when we gave back the regulatory T cells, poof, there's no pH. And then we started looking for evidence of Treg dysregulation in people with PAH across spectrum. It all started to kind of gel together. But I also recognize, I was actually thinking of Kurt's presentation today because Kurt, I think, is putting forth a model where it calls for something else happening first and beckoning inflammation. And it's not completely clear that the inflammation is the initiator. So I think there's definitely room for both perspectives.
So I'm PJ Utz. I'm a rheumatologist here at Stanford. I run a research lab and also run the MD PhD program, which I inherited from Mark. And I have to leave promptly at 10 minutes to ask the dean for $5 million for the MSDP. So you can't be late for the birth of a child, a golf tee time, or meetings with the dean. Maybe in that order. I haven't been to most of the morning session, and I actually have a couple slides from a talk that I give with an advanced immunology course about the difference between inflammation and autoimmunity, so I think it'll be relevant here. But I think one thing I would also have you think about a little bit more about is that there's a new program called Accelerating Medicines Partnership that's run through NIAMS that comes directly from Francis Collins, which is to dissociate tissue into single cells and then study the proteome as well as the transcriptome using single-cell RNA-seq and things like that Mark and Steve Quake's lab have pioneered. So that's something you should be thinking about and trying to get tissues to then be able to even ask some of these questions about what genes are activated. Are there Tregs that are in the tissue that are dysfunctional? Are there B or T cell repertoire problems with the B or T cell repertoire that might even be reflected in the blood as well as in the tissues?
So I'm Ginny Stein. I'm a clinician, sclerodermatologist, and do not do any basic science. But I've been involved in this field a long time and was really one of the early people that identified the subset within scleroderma that is at highest risk and goes on to develop scleroderma, and I think that's an area that we need to go back to find these patients early on. We all see them with the low DLCO. We showed in a morphometry study that the intimal proliferation correlates with disease duration, correlates with the DLCO. So within the scleroderma community, we have patients that are clearly at risk for developing pulmonary hypertension long before they have that. And so perhaps that should be the subpopulation of patients. And even within, to Edda's question about within the autoimmune diseases, these patients are very different. So that even within scleroderma, the patients that have a certain set of antibodies, the U1 RMP that Tom was talking about, they are more responsive both to PH drugs, the Asian population with the U1RNP, even if it's pure scleroderma, or the lupus U1RNP, the nucleolar antibody that has a little bit of interstitial disease that has primary vasculopathy. These are subsets within the spectrum, and that's an opportunity to look at that population early on before they have the very severe complication of PH.
So my name is Edda Spiekerkoetter. I'm one of the pulmonologists here at Stanford. I'm a physician scientist seeing pulmonary hypertension patients, but for the most of the time running my lab. And I became very interested in the BMPR2 signaling pathway through my postdoctoral research fellowship in Marlene Rabinovich's lab. And so I see my role here on this panel in a way to reconcile the genetics of pulmonary hypertension and the BMPR2 pathway and the changes in the immune system that we see. And so there are a couple of when you have this BMPR2 focused mind, then there are a couple of ideas that come to mind when you hear these talks. I mean, one thing, for example, when IL-6 was emphasized, you know, as a strong chemokine that can produce pulmonary hypertension by itself, then we also know that IL-6 via STAT3 can downregulate BMPR2. So I think that's kind of a very interesting connection. And then when you look at leptin and leptin receptors, and they also act via STAT3, and STAT3 gets increased. So that leads you also back to BMPR2. So I would really pledge all the immunologists to really look at how does BMPR2 or dysfunctional BMPR2 signaling influence the immune system, and does that also then, is this still kind of the basis, but has of course not only effect on the endothelial cell, smooth muscle cell, but also on inflammatory cells that we have not really paid attention to enough. So to really dissect all the different immune cells and see where does BMP play a role.
I'm Steve Abman, a pediatrician from Colorado, and you may have heard enough from Coloradans already this morning, so I'll keep this very short. But Ruben, Kurt, Norbert, Mark Nicolls, they've taught me a lot about the links between inflammation, now autoimmunity, and pulmonary hypertension. So as a pediatrician, there are a couple points I'd like to make first, and one is an NIH workshop a couple years ago where they are trying to bring together pulmonologists from adult and pediatric circles to think of primary prevention of disease. One of the targeted areas was pulmonary arterial hypertension. And we talked with such a rare disorder, how complicated and hard that is. But with what Virginia was saying, here we have a group of patients we know are at risk. And how can we get smarter and learn to identify them better with proteomics, with perhaps even informatics strategies, novel things in the clinical course or history that we haven't previously identified. So both biased and unbiased strategies from any kind of source we can get, but looking longitudinally. And I'm sure we'll hear from Paul later about, you know, RVPA coupling as an early sensitive marker perhaps, or other echo parameters and things along those lines. So I think there's a lot of excitement of how to bring this together for the prevention of such a horrific disease. Do I think there's a causal link? I'm convinced from the morning's talks and from all the tissue we've harvested from biorepository samples and the stories from the animal models that there must be. So I think there's a real opportunity here to dig even deeper clinically. The other part of it is what excites me is the idea of perinatal origins of disease as a theme, and how to link that with immunologic programming, and how to link that with risk for pulmonary arterial hypertension, and how to perhaps bring some of those things together. So this morning I was very excited by hearing Dr. Ormiston's presentation about the NK cells in the placenta and you know, preeclampsia. And certainly there's a striking story about women who develop preeclampsia with pregnancy have ongoing risk for systemic vascular disease as well as pulmonary vascular disease. And we know that newborns who've been born with preeclamptic histories, even if they're asymptomatic in the transitional period, when they're studied during adolescence, they have some elevation of RV systolic pressure by echo. They have some abnormality of peripheral systemic vasoreactivity. So there is a vascular phenotype following a preeclamptic exposure. Is it the NK cell? And somehow, are programming through that, what are the linkages through what's happening at the placenta itself? So it's one example of thinking how these perinatal events may alter immunologic response susceptibility, as well as how it might primarily alter perhaps lung and lung vascular structure. So that's it. Yeah.
And so if all of you had to think about now implementing therapies, right? Getting back to your idea of trying to find the at-risk population, or the idea that Norbert and Mark Krasnow talked about, of trying to find, what's the best population to try a therapy in, to really figure out whether this is the route to go. I wonder if we could get your thoughts about how to do.
Well, I've been trying to encourage this for many, many years. I initiated about 10 years ago an exercise study, thinking because of that old genetic study showing that the patients with familial disease had positive exercise. As it turns out, that's not a better marker than just the DLCO. They have a higher risk for some type of pulmonary hypertension, but not pulmonary arterial hypertension. And so not sure where to go, but I think that's the population that at least we have in our hands, because you're not going to get an IPAH patient beforehand. You just don't know who's going to be there. So I think that's the group of patients that are either immunological, vascular, or something, or a little bit of both. But I think that's the population you have to do.
Well, I would say one of the important things is animal modeling has been challenging, and now we have a number of animal models. I think maybe Ruben's in the back of the room, but I mean that type of model where it's so clear that you're generating a Th2 response is extremely helpful, and with a specific pathogen, and saying, you know what, we should probably go after something in that cascade in that type of pulmonary hypertension. Where it becomes more difficult when you have, we talk about this all the time, that we're still not quite sure if all roads lead to Rome. So if you can have multiple events that culminate in a form of occlusive arteriopathy, but they may be very different. And because there are different animal models that take us there, I think, well, one of the things that we're trying to do to deal with this is that for high impact pulmonary hypertension basic science papers, it's probably good to have more than one model, and to say something, it succeeds in one model, two models, three models, and what are the biomarkers that predict in that animal system. But right now, the next best step, I think, or maybe it's just as good, is actually immune phenotyping, which is what's already going on with the proteomics core under Gary Nolan with Bill and PJ. I mean, so starting off with a massive effort at immune phenotyping, and then taking your best guess. Because as we know from gene array studies, just because something's upregulated, that may be the response to
That's the question.
Right. Well, we don't know. So if it's upregulated and you have an animal model and you can reverse the disease, then it gives you a clue that you might have some hope of it working.
Yeah.
So I'll just add a little bit to that. I think the fundamental question when I approach diseases like this is to ask, is it autoimmune, or is it inflammatory, or is it something else? And so the hypothesis that we were testing as part of this proteomics core is to test the hypothesis that it's a heterogeneous disease. There'll be a subset of patients that are clearly immune-mediated and others that may not be. And within the immune-mediated, you have to ask the question, is it autoimmune or inflammatory? And that's difficult because you have to have evidence of either an autoantibody or an autoreactive B cell or an autoreactive T cell. And ideally, you'd like to be able to do passive transfer experiments in animals to show that you can either break tolerance. So breaking tolerance is super important if you're going to, I don't know how much data there actually is where someone can show that tolerance has been broken in these animal models as opposed to inflammation. And we argue about this all the time. I teach part of the advanced immunology course, and I give we have a 2-hour didactic lecture where you go over like 15 different diseases to go through the evidence of whether or not they're autoimmune or inflammatory. And so barring that, until you have evidence of that, I think you'd have to say it's an inflammatory condition. It may respond very well to various anti-inflammatories or B cell or T cell depleting therapies, but it would be ideal to know which subgroup actually have this, have the autoimmune component.
But even within the autoimmune, I mean, scleroderma is clearly an autoimmune disease, but the autoantibodies aren't the things, at least that we know of, that are pathogenic at all. So there's something even before that, or maybe the antibodies are just a total marker of not, genetics and environmental, and really don't have anything to do with the pathogenesis. So
Yeah,
it's a real challenge as far as the antibody issue.
Yeah, I agree. And people have tried pretty hard to transfer antibodies in
Yeah, it
scleroderma and
doesn't
have
do
failed,
it.
and it doesn't work. So presumably, I would argue there's probably some other B or T cell that we're just not smart enough to figure out what it is right now that's causing it. And one class to be thinking about is antibodies against either cell surface receptors or cytokines, chemokines. It's a field that's expanding dramatically. There's antibodies against things like GM-CSF that cause, is that pulmonary alveolar proteinosis, or I can't?
PAP.
Yeah, I can never, I think. Yeah, fortunately, there's a lot of pulmonologists here, so I can ask. And antibodies against EPO. We've shown antibodies against 5 or 6 different cytokines and chemokines in lupus. It's a huge field that's expanding, so I would bet anything you're going to find antibodies against specific endothelial proteins or other cell types that we're just not smart enough. It's easy to study blood. It's not easy to study these tissue-based diseases.
I've kind of another thought that I think it might be interesting to define kind of the hierarchy of the immune system that's important in pulmonary hypertension, just like all the signaling pathways that we know are important. It probably does not make sense to target each signaling pathway, but to kind of find a master switch or kind of a more pathway that's more up in the hierarchy. And so that's why, is there any cell type or any like for example T regulatory cells or so that are very high up and that can regulate B cells and all this, that we could rather target than any of the many downstream players?
Well, the answer, I think the answer might be yes, and that maybe regulatory T cells are important. The problem is that when you go up too high, then you risk immunosuppressing too much. So it is nice if you have the luxury of figuring out the best mediator for the best disease, and it's as small as possible. But on that big note, I was going to ask Ginny, were you involved with the Scott trial?
Only peripherally. I mean,
Are
I've
you-
tried to enroll people.
okay,
Maureen is here.
Maureen? Oh, okay. So a question that gets to, I think, Ross's question too is, I think Roham asked earlier about bone marrow transplantation. What happened in scleroderma with the patients with regard to when they got? I know pH was excluded, but they got the conditioning regimen versus the transplant, and they survived. What was the feeling BMT was doing?
The U.S. study results are not known because the study is still, the U.S. study, the Scott trial, is still ongoing. So we don't know. We've done some immunophenotyping, looking at patients who were on the control arm and the treated arm together. We are not currently permitted to divide those. And so I can't answer your question.
Okay.
And it wasn't done in the European ASTIS study. So,
But these were also not the patients that were at high risk for pulmonary hypertension.
Right.
So it's going to be a little bit hard to know.
I'll just add one real quick thing, though, to that, is that, so we had a patient here who had lupus who was transplanted, Sam Strober transplanted, and this patient had very bad pulmonary hypertension that completely reversed after getting the transplant, which to me was really, really surprising. I would have assumed it would have been fixed. Again, these anecdotal things can argue that there might be an important T or B cell response or something in the bone marrow, whatever it is, probably B or T cells.
Well, we were hearing rumors, and again, that the conditioning regimen was having an impact. And I am aware of some cowboy rheumatologists at other institutions that are doing very aggressive cytotoxic agents, very aggressive, and seeing results. And this always bubbled up in the conversation that they're seeing similar results in the conditioning regimens for the Scott trial. So, and again, that's making the assumption that if people start with scleroderma, whatever's causing scleroderma, if they evolve into pulmonary hypertension, it seems like it's the same spectrum of the disease process. So that's the leap. You know, that does, and it doesn't seem like that big of a leap, but I don't know.
I don't know. Yeah, it's on. It is a comment, and it is in terms of trying to address the issue of how the disease develops and how it interacts the pulmonary vessel with the immune system. So we know plenty of examples that inflammation in the pulmonary arteries by itself doesn't cause PAH. There are several, Wagner's granulomatosis and even PAN, although it's rare in the lung, it doesn't cause PAH. In pulmonary arteries are involved as bystander in alveolar inflammation. It doesn't cause PAH. The other aspect is that you have several forms of lung inflammation that by themselves are also not associated with PAH. Many cases of rheumatoid arthritis that is incredibly inflamed, the lung is not sufficient. So there is something that's a holy grail between the vessel wall, and I'm going to call immune privileged, but with a lot of caveats. So there is a tolerance in terms of the immune system and the blood vessel function that we don't understand how this communication happens. So it has to break in one point. I think that one conceivable is that there is a vascular injury that is very specific to trigger this break of the immunotolerance. And therefore, the next step has to be either innate or acquired immunity or both, or a sequence that we don't understand. And until we can start linking these two events, it's going to be incredibly hard to really to making sense what is happening and how to affect in terms of therapy and subphenotype the patients. I don't think that's unlikely BMPR is going to explain it all. I wish maybe it's true, but unlikely in my point of view. But nevertheless, it gives us a really important hook because at least Nick Borrell in a Nature Medicine paper a couple months ago shows that BMP9 rescues the vascular integrity. So it gives an opportunity now to maintain the integrity of the vessel and see why this inhibits the recruitment of inflammatory cells and the downstream events. So I really think that this is one of the difficulties is to really decipher what is involved in the vascular injury and recruitment of inflammatory cells in innate. And I think that collagen vascular disease is another way to start addressing that because it has to be what you said. Virginia said that something precedes the autoantibodies, and I think that we need to start understanding that. If not, we are not going to be able to make inroads in terms of cause-effect relationship.
Oh, just start.
Does that work? Yeah. I'd like to go. I wish I had a flip chart because I want to draw a diagram, but see if you can visualize this. And I'm talking about scleroderma now. Scleroderma, the typical is, we're talking about a 20-year disease. 10 is under the radar. 10 is with Raynaud's, or 8, let's say, is with Raynaud's. And then the last 2 is symptomatic and progressive pulmonary hypertension. So I think we're talking about what's going on in the last 2 years of a 20-year disease, which is very complicated. There are multiple mechanisms. There are probably reparative and damage mechanisms that are going on. And I'm wondering if we're going to look at scleroderma, if we would be best. I'm not saying we shouldn't do the treatments that Norbert is suggesting. I'm saying that empiric sort of activity really should go on. But from an investigative point of view, I wonder if we shouldn't go look at the RA model, which is pre-RA. You can't call them patients because they're just subjects, but they are first-degree relatives of patients with rheumatoid arthritis who have rheumatoid factor and anti-CCP antibodies. They have abnormal HRCT scans of the lungs because they have some kind of peribronchial abnormality that those first-degree relatives who are antibody negative don't have. Their BAL fluid, even if they're antibody negative in the peripheral blood, can have rheumatoid factor and CCP in the BAL. So I'm wondering if we shouldn't go even further back than what Ginny suggests to try and develop a multi-center cohort of individuals who are at risk, and they would be in some way first-degree relatives. Another possibility is because of the high numbers and the increased frequency of pulmonary hypertension, anti-centromere positive Raynaud disease patients who can't yet be called scleroderma. And I wonder if we shouldn't be thinking that far back, that far upstream, if you will, to try and develop cohorts for investigation. Now, the pulmonary arteries may be a problem, but the same types of things are going on in the digital circulation, and maybe the skin tissues, the digital tissues with their microvasculature would be amenable to that, to investigation. So I'm raising that question.
Yeah, it seems like it's calling for a new mechanism of NIH funding, right? Or something along the lines of
20-year disease.
the cardiovascular Harvard, Framingham. I mean, so you really need something set in place for a decade before. What is the prevalence of PAH? If you're diagnosed with scleroderma, what are the chances that you will evolve into PAH? 11. Right. So.
But that's probably an underestimate because we don't have people who are followed until they die from something else. So
right. So you'd have to treat.
Some patients are going to accrue the disorder, and we say 70% in a cohort that's had about 60% have had a 5-year follow-up after their first visit.
We could ask Paul Hassoun to comment on the results of his early intervention. I guess you'd call it is a trial, right, in scleroderma?
I should say that the Dutch now have an intervention trial in RA, pre-RA, with antibodies, Rituxan versus placebo, for prevention, delay of progression to clinical RA.
So I was not going to comment, but there have been several interesting comments here. First, in terms of inflammation, this is a symposium on inflammation. I'm starting to think about this disease more of a disease of the fibroblast and the vessels. I think at the time we see these patients with pulmonary hypertension, I'm talking about the scleroderma patients, I think they have very little inflammation and autoimmune problems are probably not playing much of a role, but what is striking is the fibrosis, whether it's the skin or other organs, the lungs. We've been particularly interested in why these patients don't do as well as patients with IPAH, and we've been comparing those groups for the past 10 years now, those two groups. And now it's clear that they have other problems. For instance, they have myocardial intrinsic disease. They have, even when they don't have PAH, the myocardium contractility is significantly decreased. And I think this is directly related to decreased myocardial perfusion, which we have shown by adenosine MRI, a stress test. So it's a very complex disease. It's also the interaction between the right ventricle and the pulmonary vascular disease. Now, until a few months ago, I thought that we had no treatment for these patients, and because the median survival was like 4 years, pretty terrible, compared to 8 or 9 or 10 years for IPAH. You asked me to comment on the very small trial that we just completed, thanks to Gilead and United Therapeutics, where we gave a combination of ambrisentan and tadalafil. And I've never seen this in the past 15 years. Some of the patients with severe pulmonary hypertension have normalized their pulmonary vascular resistance. Roham, actually, Stanford was involved with this, Cleveland Clinic, UT Southwestern. So, I mean, it took several centers to recruit just 24 patients, and this took over 5 years. But this is the first time that we see a treatment. I'm not sure what happened with this treatment, whether there is some antifibrotic effect of this combination, but we have patients who have almost resumed a normal life, which I've never seen before.
And what
So
was
that's
the
all.
entry? How mild was their disease when you started therapy?
So we just wanted a PVR greater than 3, mean PA pressure of greater than 25. So there were patients with PVR of 12 or 15 and others with PVRs of 3 to 4. But many of these patients, I would say maybe 1/4, normalized their vascular resistance.
So I mean, it's a little bit of what was being gotten at with the question of a longer-term study. But do you think it's possible then that inflammation can contribute to the beginning, but that the high pressure head exacerbates and you have a positive feedback, so that the higher the pressure, the worse the inflammation, or?
Well, I'm not sure. I think I learned from Ginny a few years ago that there is not much inflammation at the time we see these patients. And what is striking is really the fibrosis and loss of vessels, whether it's in the fingers or in the myocardium or in the pulmonary vessels. Now, several years ago, Ruben was interested in comparing IPAH to scleroderma in terms of, it's clear that they don't have plexiform lesions, but I think there is much more loss of vessels in the lungs, and this is what the DLCO is significantly lower in these patients compared to IPAH. I'm not talking about patients with any degree of fibrosis. We try to exclude those, but although you can't, I mean, they all have some degree of fibrosis. But so this is why I'm thinking about this disease as a disease of the fibroblast. This is why I asked Kurt Stenmark whether he had done biopsies of, I mean, this is easy tissue to analyze in terms of what you can do with fibroblast metabolic activities. So I'm not sure I am answering your question, Mark, but...
That's good. I think, do you want to just, can we end with your...
Before lunch. A question that I have is, what happens in those rare, unfortunate individuals who are both BMPR2 heterozygous and also have an autoimmune disease? You would expect that if these two factors are interacting in the same disease, you would get a very fulminant form of PH, early fulminant, in individuals who unfortunately are both heterozygous for BMPR2 and also, are there patients like that? Are there individuals?
So in scleroderma, they did not find an increased association, but whether there were individual patients, I don't know.
Mark? Yeah.
Yeah, we have a study in France with a cohort of patients carrying a BMPR2 mutation, but pre-symptomatic, and they are in a regular yearly screening program. So none develop an autoimmune disease, so it's not the case. But what is interesting, I can just share with you the 3 first pulmonary hypertension cases, which are incident in the setting of BMPR2, all in females. That's interesting, but it's only 3. And second, in young females, always after pregnancy. So is that the shear, the volume load, the stress on the endothelial cells, etc., or is it something immune? So we have, of course, a biobank, and we will test it later, but that's interesting. I think the beauty of BMPR2 mutants versus scleroderma is that it's exclusive. These people are doing fine. They have no systemic conditions. They are exclusively they are normal, healthy people carrying a mutation. So that's interesting. It's complementary to scleroderma, I think.
So the patients that Grunig did the exercise study,
Yeah.
did any of those patients go on to?
Yeah, in the study, they have to volunteer to many, many investigations. And it also includes, at baseline, only in volunteers, an exercise hemodynamic testing. So they are not complaining of anything,
Right.
but we do a
It was
right
just-
at cath
right.
with exercise. And all of them at baseline have normal exercise hemodynamics. But all of them volunteered. That's interesting to know.